Welcome to the Jack Westin MCAT podcast with your hosts, Michael Maleka
and Molly Hudash.
Hello, everyone, and welcome back to the Jack Westin MCAT podcast with your hosts, Mike and Molly. Mike, this is going to be a really fun episode and a super high yield episode, right? We're coming from a discussion of endocrine system, right? All about this long-term homeostatic balance with our hormones. Now we're talking a lot snappier, right?
A lot faster, right?
Shorter term.
Right.
What are we talking about?
We're talking about the nervous system today, right? And this is again, like you were saying before, really great for after the endocrine system because we talked about the endocrine pathways. It needs the blood to actually go, but in order for it to travel in the blood, it takes the blood travels quickly, but not that fast, right? We want to be able to respond to things really quickly. Like if I see a bear, I don't want to wait, if I see a bear, again, we learned how to deal with bears last episode, right?
Yes.
Wondering we could watch back that one.
Yes.
If I see a bear, right, I want to be able to respond immediately. I don't want to wait 10 minutes for my cortisol to get to where it needs to go. So this is where our nervous system really kicks in. We need to be able to respond quickly to stimuli. And this is where our conversation of the nervous system begins, because we want to be talking about the different parts of the nervous system, right? Where there's kind of different areas, different things, or different parts and serve different purposes. And I think this is kind of, again, like you were saying before, that high yield material, because the MCAT expects us to know, right, what parts of the nervous system are responsible for what and how they all really work together.
Yes, for sure. And this section, right, or this subject of the nervous system is incredibly important because this is really, and we were talking about this earlier, this is really the only topic that can be tested in any science section.
Which is so cool. Right?
The nervous system itself is obviously the foundation of psychology, right? That's the biological basis of behavior. You're going to get a lot about neurons and action potentials, and then the different important structures of the brain and the spinal cord and nerves and all that good stuff. Very much psych-based. Of course, the nervous system is also very testable in biochem. It is a biological system.
Right.
And it is also very easy to apply to chemistry and physics.
Right. When especially like circuits, right?
Circuits, especially, yes.
Right.
Because ultimately we're talking about the flow of charge, which we'll get into in a much
eventually one of these days.
One of these days. No, later in this episode. But a lot of the nervous system content can be discussed in chemistry and physics as well. I would say nervous system is probably the highest yield body system on the MCAT, and honestly, it's a big statement, and I'm
a big statement. I have to think about that. Yeah.
But it's in every section. It may not be the highest yield in biochem, but if you think of the 230 questions that you're going to see, or the 230 minus 53 that are science, I think it's probably the highest yield body system.
Interesting. I've actually never considered that before, but I mean, I get the thesis, right? Because you're like, it could be in any section, so technically it could be tested the most. No, I like that. I like that. Yeah.
Testable in all the sections.
Right.
Like it's not like, oh, it's like you could maybe talk about it in these sections. Like it's very fair game in all three.
It's very fair game. And again, that's just even more reason to listen to today's podcast, right? You're here with us. You're going to learn about the super high yield system.
Yeah. And it's crazy because initially, we mentioned this when we were first introducing this content-based segment that we're doing, or series on the podcast. We sat down and mapped out we taught this. What is the next logical step? What should we teach next? What should we teach next? And they all build into each other. And we've been on this homeostasis, workflow. We actually were initially going to go to a different topic after endocrine. And then we finished endocrine and we're like, how could we not talk about the nervous system?
We can't. We literally, it just fits so perfectly after.
And it's so high yield, right? We adjusted our initial plan to include this. So this is kind of a special, it is homeostasis. We're going to get back to our homeostasis focus after this episode, but it's so incredibly high yield.
Right.
And it's so important to talk about this after endocrine, right? So endocrine, go back and watch the episode, right? It's all about hormones traveling through the bloodstream, very long-term regulation of growth and development and metabolism and all of those things. That being said, if you're being chased by a bear, you need to respond fast. So in our discussion of the nervous system, I feel like it's important to start with just, what is it, right? We'll get into
a great place to start. Like, what even is this thing?
Right, like what is it meant to do? And
Right.
what are the different components of the
Mm-hmm.
nervous system, not a neuron,
Mm-hmm.
the nervous system,
Mm-hmm. Mm-hmm.
right? Let's start there.
Right. I like where you're starting. Like, okay, let's get a broad overview. Because when we
Yes.
think about the nervous system as a whole, we can first begin to separate it into kind of two different kind of not again, I hesitate to say structures, but two different type like parts of the nervous system,
And
right?
division.
Segments, divisions, sure, yeah, that's a better way. The central nervous system and the peripheral nervous system. So again, generally we start at the central nervous system because again, I know it's central, right? When we talk about the central nervous system, this is when we talk about the brain and spinal cord. All that electrical activity in the brain and spinal cord, all of those kind of those neurons that are associated there, anything that's located in the brain or spinal cord is part of the central nervous system. While all of those sort of neurons on the outside, right, on the periphery, outside of the brain and spinal cord, are part of our peripheral nervous system, right? And it's kind of this is kind of the first main division, right? And when we talk about the central nervous system, right, like I kind of want to kind of first talk about that. We think of the brain, this is the major processing place. This is again where so much, this is again, we're not going to dive into the different lobes of the brain today. We're not going to talk about that, but that's such a high yield area where a lot of the processing, your sensation, your conscious, we believe, is in the brain, right?
Mhm.
So, but the spinal cord, a lot of times we associate it with just being a tract, with being a tract like that connects the brain to the rest of the body. But I know, Molly, we were actually talking about before, that's not the only, like it's not just a pathway, is it?
No, it definitely isn't. And it's so funny because I feel like even in studying for the MCAT, we focus so much on the brain, pretty like not exclusively. We talk about nerves, we talk about, the spinal cord, the different, layers of the vertebrae that correspond to the spinal cord. Yes, not necessarily anatomy level, but we talk about it. The brain we talk about in so much detail. And I feel like we are geared to assume the brain is responsible for everything.
Mhm.
The spinal cord, the peripheral nervous system is like secondary. It doesn't matter as much as the brain. And yes, brain is important, but the spinal cord is awesome. Sometimes the spinal cord can actually operate independently of the brain,
Absolutely.
which is what we're kind of talking about when we talk about reflexes.
Hmm.
So peripheral nervous system, we didn't talk in a ton of detail about, but those are the different neurons that come off that spinal cord to create sensation, right? Which would go up through the spinal cord to the brain, and also those signals that are motor that are coming from the brain back down through the spinal cord and out, right?
Mm.
When we think of, let's say, touching a hot stove,
Mm.
right? Obviously, that's going to create a sensation of pain and
Hmm.
burning. It's going to kind of stink, right? We
Right.
don't like that. It's going to be painful, right? That sensation is going to be carried all the way up to our spinal cord by our sensory neurons,
Hmm.
obviously carrying the sensation of pain with our
Hmm.
nociceptors. Um, and normally, right, if this was, a light touch or something, kind or like neutral, it would go up to the brain. The brain would integrate it, bounce it around a little bit, decide on how I want to respond to it, and then send it back down to the spinal cord, out through motor neurons to respond, right? If it's pain, the spinal cord can operate independently of the brain.
Hmm.
Signals still sent up to the brain, but it's slow, right?
Hmm.
We don't want that painful stimulus bouncing around in the brain too long. The interneurons in the spinal cord, which we'll talk about, can actually make a decision. Yes, brain, you will hear about this, but first, please get your finger off that stove,
Hmm.
right? It itself in the spinal cord can make the decision to
Hmm.
send out a signal through your motor neuron to move off the hot stove.
Hmm.
That being said, right, if we didn't think it went up to the brain, you'd have to ask yourself, Mike, what would you do? You just stuck your hand on a hot stove. Yes, you
Hmm.
move, but you're probably gonna do some other stuff too. What would you
Right.
do right after that?
If I like first, if I like if again, if we're talking about like after
Like,
that,
ow,
I at first I my hand would move before I even felt the
before
pain.
you even felt the
Before I
pain.
even felt the pain.
Mhm.
But then I would think, then after my hand moves, I'm thinking, oh, that hurts, right? I should probably do some different things, like
Yes,
turn off my stove,
off the
right?
stove.
Like get this pan like away from me kind of deal, right? Why did I even touch this in the first place? I've
Right.
learned. But a lot of those things again happened as a result of the processing in my brain. But imagine if we didn't have this, right? That was so slow. if we didn't have this reflex, my hand would have been there the whole time until I've decided that, oh, this is painful,
Ouch.
right? Like,
Oh,
is this
okay.
painful to me? Right?
Dude, your hand
Right?
is fizzled off.
It's in my hands like all off now. So this is kind of really outlines the importance, like you were saying before, of the reflex, where again, it takes time to go up to the brain, especially again, not all reflexes are just the hand. Like we have knee reflexes and the rest of the body, where if it takes that time to get up the brain, damage could occur unless we act quickly. This is the importance of that reflex, 100%. And again, really speaks to this idea that, again, the spinal cord isn't just this pathway between the peripheral nervous system and the brain, right? It serves a function, especially with reflexes, which I think super cool.
Yeah. And you will learn a lot about the spinal cord with medical school when you go really in depth in A&P, anatomy and physiology. You just don't really need to know that much about it for MCAT, right? For the most part, spinal cord testing on the MCAT, it's in the central nervous system and it's responsible for reflex arcs.
Yeah.
That's pretty much it. So we
No, that's
wanted to
awesome.
include it and also just to give a little love to our non-brain components of the nervous.
Give a little love to the spine. The spine
Give
gets
a
a little
little love to the
neglected,
spine. The
right? I love
spine
that.
the spine works so hard to keep us upright and also
Right.
to do pretty much everything else for us, right? Other than complex thought and emotion,
Right.
right?
Yeah, I don't think the spine does anything of that, as far as I know.
I, who
But who
knows?
knows? But I think
But
I love these divisions because CNS, I think, in terms of divisions, is just the brain and spinal cord, which I actually really like. I appreciate that about the central nervous system. But then when we talk about the peripheral nervous system, there's so many subsets. Like, okay,
yeah,
there are so many ways that we can deconstruct the peripheral nervous system. So
because
let's
it's
kind of
really everything else.
it's basically everything that's not the brain and spinal cord is the peripheral nervous system. And again,
Exactly.
there's so many functions in the body that we have so many different types of nerves and classifications and structures. So the first sort of like main division of the peripheral nervous system is a difference between the somatic nervous system and the autonomic nervous system. And that's kind
Yes.
of the first main division. When we think of somatic, right, the nervous system, again, remember, we're still in the peripheral nervous system. When we think somatic, we're thinking about conscious motor control. We're thinking of moving, again, the somatic neurons moving my hand. The fact I'm waving my hand right now, for those who are watching on YouTube, if you're not watching on YouTube and just listening, I'm waving my hand right
Spotify.
now. Or on Spotify, if you're watching the video, which, hey, I'm moving my hand right now. I'm moving my mouth right now. There are nerves that are responsible for contracting my muscles and having this conscious control. Those are our somatic motor neurons or somatic neurons. This is very different from the other like kind of class of peripheral nervous system, which is the autonomic neurons. Right? These autonomic neurons, kind of the main difference is that I kind of think about them as automatic.
Automatic.
Yeah,
Yep.
like right? Where it's like I don't have control over the autonomic neurons. They sort of function, right, in response to stimuli, in response to keeping, sort of homeostasis, right, in response to like different stimuli that I'm not in control over, like the idea that if I'm scared, my heart changes beat. I don't consciously control that. And that's kind of a good kind of rule of thumb. Like if I don't consciously control it, it's probably part of the autonomic nervous system. If I consciously control it, it's part of the somatic. Right?
Yes, absolutely. And when we further classify the autonomic nervous system, you're
Mhm,
going to see words like stress being used, right?
mhm,
Autonomic nervous system, again, is automatic. And the primary divisions that we talk about
mhm,
in an MCAT sense are the sympathetic nervous system or the fight or flight as you hear it,
mhm,
which is basically a short-term stress response.
mhm,
And then we have parasympathetic, which is the rest and digest, which basically is I'm not super stressed.
mhm,
And you're gonna see diagrams that will show you all the different functions of these. But I feel like it's valuable to consider short-term stress. Let's consider sympathetic. These two
mhm,
pathways are pretty much exact opposites of each other. So
mhm.
when you walk through a diagram like that, I don't want you to memorize. It's really
Please
hard.
don't.
Just
Yeah.
imagine, right? Fight or flight is active. Let's say when you see a bear,
Of
that's
course, we got to refer back to the bear.
right. But it's also, let's say, if you are doing public speaking, which
Hmm.
for most people that aren't, weird like us, that's
Yeah.
uncomfortable. You would
I live
probably
off
have
this.
sympathetic. Yeah, I mean, it's maybe more applicable than seeing a bear in the wild.
Correct. Correct.
Most people have been, had stage fright or
Mm,
fear of public speaking or fear of something, right?
mhm,
You're in a situation where, oh my gosh, all of a sudden, this is scary,
mhm,
right? I'm that feeling of panic that's immediate. not
Mhm.
long term, like cortisol signaling with endocrine system, immediate set in, right? Your heart starts to beat faster. You start to kind of breathe faster. You
Mhm.
start to sweat, right?
Mhm.
You start to kind of anxiously look around the room. You're paying a lot closer attention. These are all things of the sympathetic nervous system, right? We
Right.
talk about how your pupils dilate.
Mhm.
Why, right? Pupils are letting light in, right?
Mhm.
You're basically a lot more in tune with your visual environment when your pupils are dilated. You're getting a lot more light coming in, which
Mhm.
carries information about your surroundings. Again, your heart is going to speed up, and
Mhm.
your liver is also going to help increase the blood glucose level, right, that you have floating in your blood, right? The whole idea is get glucose, get energy to your skeletal muscles and to your brain. Those are
Mhm.
the most important two if you're going to fight or you're going to flee, and you have to
Mhm.
decide which one, right? You're going to change blood
Mhm.
distribution in the body or the blood flow to the body. You're going to prioritize systems like the brain, the heart, the skeletal muscles. You don't need as much to the digestive tract right now,
Yeah.
right? We're in a
Yeah.
fight or flight situation. I don't care if I've gotten every ounce of, like dietary protein from
Mhm.
the food traveling through my GI tract right now. That's
Mhm.
too long term. I don't care about that right now. I'm about to get eaten alive,
By a
or
bear.
I'm about to pee my pants in front of a giant audience, which, by the way, you do have relaxation of your bladder in your
Mhm.
sympathetic nervous system, which is why if you get really scared all of a sudden, in extreme
This
situations, some
happens. Mhm.
individuals will
Mhm.
urinate, right? Because
Mhm.
the bladder relaxes.
Mhm.
All of these symptoms of sympathetic activation can be thought about as put yourself in that situation.
Right.
Again, you're going to sweat. You're going to sweat. And
Mhm.
if you are sweating, it's primarily because you're trying to absorb heat from the surface of your skin in anticipation of running or fighting or doing
Mhm.
whatever, right? Muscle contraction creates a lot of heat. You're trying to cool yourself off in advance. If you're not moving, you might get the chills. You might feel anxious,
Mhm.
and you don't have heat to lose, but you're prepared for it. Again, keep your mind on the fact this is very short-term response to stress.
Mhm.
If we know what sympathetic does, parasympathetic does the exact
Exact
opposite.
opposite. Mhm.
I'm not stressed. I
Mhm.
don't need to increase my heart rate. I don't need to
Mhm.
sweat. I don't need to breathe faster or harder. I don't need my pupils so dilated. I don't need to shunt blood flow away from my GI tract and reproductive organs. I
Mhm.
don't need any of those things. I
Mhm.
don't need to increase my blood glucose level. I'm good where I'm
Mhm.
at.
Mhm.
Um, so that those are the main distinguishing characteristics to
Mhm.
kind of refresh central nervous system, brain and spinal cord, peripheral nervous system, right? Everything else.
Everything else.
Everything else can be subdivided primarily into somatic, which are,
Mhm.
different types of nervous tissue that are under voluntary control, and autonomic, I almost said
Mhm.
automatic, right,
Mhm.
which includes the parasympathetic and the sympathetic nervous system.
Mhm.
Is there anything that I missed, Mike?
I think like I would maybe just make like the only thing that like I think we like talked about this before, but like the motor neurons, like when we talk about like the
Yeah,
autonomic and sympathetic nervous system, there's only motor neurons
yeah.
in those, and like the sensory nervous system is again because it's in the periphery, like my terminals, like the nociceptors, they're not in the brain and spinal
Detect pain,
cord.
by the way.
Oh,
I
that
threw that in there
sure.
without
Yeah, yeah.
clarifying.
You threw it in earlier, so I was building off that. But yeah, nociceptors are the nerves that kind of detect like pain stimuli, and they're not in the brain and spinal cord, so they are part of the peripheral nervous system. But which bucket they fall under the peripheral nervous system? A lot of times, it's like, oh, where do they fall under? Sensation or some sensory nerves fall under the somatic nervous system. So yeah, so all motor neurons, like you can have motor neurons that are under, the somatic and the autonomic, but sensory neurons are just part of the somatic nervous system. And so the reason I think this is important is because, when we talk about the parasympathetic sympathetic, all of those are motor neurons. Like
Yeah.
you have no sensory neurons in the sympathetic parasympathetic. It's all just, oh, I have this stimuli, I see the bear, things are happening. I'm not sensing things differently.
Yeah.
So
Yeah.
that's
And that,
probably the only thing I'd add. Yeah.
yeah.
Mhm.
And I feel like this is a good time to build on this because we're saying Neurons, neurons, neurons.
Ah, yes.
I
We've
feel
said it
like,
a million times now.
yes. And hopefully this is a term that you've heard at some point before. Right? A neuron is an individual single cell of the nervous system. It's
Yeah.
kind of like when we talk about a muscle,
Hmm.
right, is made of a bunch of individually functioning units, right? Contractile units called a sarcomere, even though there are actually many sarcomeres in one cell, right?
Right.
It's kind of the individual functional unit of a muscle,
Mhm.
or a
Mhm.
nephron is the individual
Right.
functional unit of the kidney. A
Sure.
neuron is the individual functional unit of the nervous system. In
Uh-huh.
this case, it is an independent cell, not
Hmm.
just a structure, it's a cell. And it's important to note, by the way, that this is a cell. It has all the
This.
normal cell stuff, mitochondria, ER, right?
Mm,
Ribosomes, all of these things.
mm,
That being said, if we think of a cell as like a sphere,
mm.
most cells aren't spherical, but you know what I mean.
Yeah.
A circle, like we would normally see in a two-dimensional drawing, it has that structure. It's called the soma or the cell
Hmm.
body,
Hmm.
but there are lots of little cellular appendages
Right.
popping off,
It's a little
right?
bit of
And
a weird one. A little bit of a weird one, that guy.
a weird shape, yes.
Hmm.
We've
Hmm.
got little tiny like fingers, basically. I
Hmm.
almost think it almost looks like a hand cut
Kind of
off.
like, I have never thought about it. Kind of like if I raise my arm right now, like this is kind of like the soma. Fingers are kind of like the dendrites, and like the
This is the
forearm.
axon.
That's a really, I've never, I like that. I like that a lot. Yeah. Yeah.
Yeah. So we got the fingers, right, which are the
Hmm.
dendrites. We've got the soma, that's the hand, and then we've got the axon, right? And we talk about the wrist as
Hmm.
kind of the what we'll talk about this in a moment, but the axon hillock, it's
Hmm.
basically the place that we go from the soma itself, the
Hmm.
cell body is also what it's called,
Hmm.
into the axon.
Hmm.
Now, to give you some context, we'll go through action potentials and synaptic transmission in a moment, but the fingers, the dendrites are all about receiving incoming signals,
Hmm.
right, from other neurons. Keep in mind,
Hmm.
these neurons are essentially relay racing information,
Hmm.
right? They're passing the baton. I am active. I want to activate you. Will you take the baton and run with this? I don't
Hmm.
know, right? The dendrites are where you receive the baton, right? The soma, we are going to take all these incoming signals and we're going to make a decision at the wrist, right? Which is
Hmm.
the axon hillock. The
Sure.
axon itself is where the majority of the transmission is going to happen. If we
Hmm.
fire an action potential, that's where it's going to propagate along. Um, and then we're going to end that axon at the axon terminal, in which we will basically pass the baton off to the next neuron.
Hmm.
That's kind of the basics,
Of the
right? We
structure
do
of a neuron,
have arms,
sure.
yeah.
It's
So,
again with lack of visuals. Again, there's all these times where it's like, man, I wish we could have like a picture and like have like a
I
neuron
know,
on
I know.
screen.
But we all have arms,
With
right?
all arms, yeah, just look at your arm. But
Well,
no, I think
probably. If
yeah,
you don't have arms, that's okay too.
that's okay. But again, when we have like talk about like the neuron structure, I think it's also important to talk about like supporting structures of the neuron
Yes.
as well that are really important for its function.
Yes.
This is where we talk about like the oligodendrocytes and the Schwann cells. And we talk about these cells, like if I ever hear this, like see those words on like in a passage, right? What should the immediate thought be, Molly, if you see those words?
Fun.
Myelin,
Fun.
100%, like myelin, 100%, right? That should be the first thought because myelin is an important supporting structure of the neuron. Again, it's not actually like part of the neuron cell, but it kind of like wraps around the terminal. Imagine if I had like a whole bunch of wristbands around my forearm, right, of the axon, right? Myelin wraps around the axon, right, and it serves this extremely important function of insulation. Right, we're going to talk about the action potential in a little bit, but again, a key thing to know about myelin is that it allows for a much faster and more efficient propagation of the signal. Right, because axons, that's what they do. They send the signal, but myelin makes it so that signal again is goes way faster, like significantly faster, but also maintains the signal strength throughout. Right, so myelin is a extremely, very important structure for sending neuron signals, and it's really bad when we kind of lack the myelin, isn't it, Molly?
Yes, it is. And
Hmm.
so we could talk about a clinical application, we will in a second, but I totally forgot until right now a really interesting, weird, like evolutionary biology thing
Oh
that
no,
happened.
not this again.
Yes, yes. Right. Again, if you haven't heard Which episode was that in about the tetrapods?
The tetrapods. Oh, what were we talking
Oh,
about?
respiratory system.
Right,
Respiratory.
because we were talking about the lungs and the yes, yes,
Yep,
yes,
yep,
yes.
yep, yep. So for context, I've been a teacher for many years and I taught evolutionary biology
Mm,
for 2 years. Um, Periods of the year was part of the Gen
sure,
Bio
sure.
series, but loved it. And when I was learning about myelin, my
Mm-hmm,
evolutionary biology professor brought up how important myelin is. Again, myelin is all about insulating. What
mm-hmm,
we'll talk about, it's basically current, think a wire, right? It's basically the
mm-hmm,
plasticky, rubbery coating
mm-hmm,
of a wire that you would
mm-hmm,
plug in your phone. It helps insulate and it also helps things transmit very fast. There are animals in the animal kingdom that do not have myelinated axons, very
mm-hmm.
specifically the giant squid.
I knew you were gonna bring up the giant squid. I knew it. I was like, I knew you were
The
gonna bring up the
gi-
giant squid.
the giant squid has unmyelinated axons, and it's a giant squid. These axons are very long, very long. Um, and that's a problem, right? Because the whole point of an axon is to insulate and help it move faster, and
Mhm,
also stop this from being an incredibly energetically expensive process. So
mhm,
the way that the
mhm,
giant squid will do this,
mhm.
actually by having axons that are huge,
Huge,
and
massive.
it's it's wild.
Hmm,
It's wild how big these things are,
hmm,
especially in relation to a normal
hmm,
axon, right? Way, way, way larger. And the idea is you have a lot more essentially insulation.
hmm,
Because it's so thick, you actually have a much smaller surface area. The bigger
hmm,
you are, the smaller your surface area.
hmm.
Well,
Oh, wait,
surface area volume ratio.
right.
Um, and they're actually able to transmit at pretty high rates
Mhm.
because of how big they are. Um, I know that this is not MCAT testable. It's just cool,
It's
right?
just cool, and I think it's worthwhile to talk about. Yeah, no, and they're actually because they're so big. Like, and like I like when I was in like I when I remember I took a class all about the like I took a class just on the action potential. That's all this class was about. Giant squid nerves are actually used for studying a lot of times action potential because they're so big. They're a lot easier to kind of study those graded potentials. Um, and
And
so.
for size context, just so you know, a human axon is on the scale of micrometers, very, very
Mhm,
small. A giant squid axon can be as big as 1.5 millimeters, which is insane, meaning you could
right,
see it. Think millimeter on a ruler, right?
mhm.
You could see this. Like
That's huge
it's
for a cell.
macroscopic. It's so
Right.
large.
Again, yeah,
That's
I think because I
humongous.
I think when you hear the word millimeter, you think, oh, that's still tiny. We're talking about a cell here.
It's basically
Cells are tiny normally. That you can't even see cells.
it's the width of a human hair.
Right. That's a nerve. Right.
Wild.
Wow.
Anyways,
Yeah.
I digress. We'll talk
Hmm,
about the clinical application because it's related to human beings, but I
hmm.
I had to mention my giant squids.
No, no, I'm on the same page. I think that's super cool because the giant squid, again, is a really cool nature adaptation to
Mm,
lack of myelin, right? Because there's kind of two things that impact the speed of propagation, size of the neuron and whether it's myelinated.
yes.
So imagine like, I've actually exactly seen this. This is actually, I think the concept is very MCAT testable. I've seen a question that asks which is the fastest neuron, and you're going to be looking for something that's big and something that's myelinated. So, you know what? I actually think it is very MCAT testable. You know what?
Yeah,
Thank you, Molly, for bringing
you're
that
welcome.
up. But
We could even think about like fluids almost and talk about the radius and flow
um,
rate, right?
I was actually kind of expecting you to say. I was like, oh my goodness, this is a great flow rate. Poisson's law. Where are you?
I
Right? Like,
know.
right.
Look
So,
at
all
that.
right,
It
it's all
all comes
connected.
together. It's
It's
all
all
coming
it
together.
comes full circle.
Yes, makes sense.
But
In
I'd. Mhm.
in the context of the human body, we do not have the opportunity to change our myelin, or we don't have the opportunity to change our axon size,
Yeah.
right? One of the things that we should mention, right, is a lot of these neural pathways, or I shouldn't say neural pathways, a lot of these neurons,
Hmm.
right, do not divide. They don't
Yeah.
divide, they don't grow, right? You
Hmm.
have the ones you have, protect them, please, right?
Hmm.
Because they
Keep it
don't
brain. Yeah.
typically divide, right?
Hmm.
They're not very active in terms
Hmm.
of mitosis. So if we damage our nerves or our
Hmm.
neurons that make up nerves, that's a problem. And one of
Hmm.
the most significant ways to impact this, other than, getting in a trauma that disrupts an axon, et
Sure.
cetera, is actually demyelinating diseases. And
Mm.
common example is multiple sclerosis,
Yeah.
right? This is an autoimmune disease in which the immune system is attacking oligodendrocytes in the central nervous system and essentially reducing the myelin on neurons of your brain and spinal cord. This has a whole host of issues down the line, very, very like dramatic in its effects, right? And very system wide, right? You could have fatigue, you can have numbness, you could have paralysis, you have balance issues, tingling, you know, it can mess with your vision. You may not be able to move, right? Very, very impactful on the body, right? You need it. And it's important to think about because it sounds like a peripheral issue because you're going to experience a lot of symptoms in your body itself, not just your brain, but it is demyelination. That's all that's happening. We're attacking those oligodendrocytes and it's having systemic effects that progressively get worse and worse over time.
Yeah. I think it's in recognizing, I think we learn a lot from from pathology, right? When you have a lack of something. Actually, I learned so much. I remember when I was in school, like from histology, when you would look up like how cells change in certain pathologies, it can really make you like really appreciate the functions that these cells serve. Like, oh, when the myelin is being destroyed, what's the impact on the body? Wow, the myelin has such a huge impact. So yeah, I think in studying these pathologies, we can learn to appreciate the functions of these things. I know in psychology, especially, like that's mainly how we study the brain, is we study patients who have, who maybe be like atypical, right, in the neurology, and then we can learn based on their deficits what the different area of the brain does. So again, we can study these things to learn about it, but yeah, absolutely, the damage to myelin, showing how important it is, has huge, huge impacts.
Yeah. And as far as why myelin is important, I feel like it's important to now address the elephant in the room,
Right.
which is the action potential,
Oh, I thought
right?
you were gonna say the giant
How,
squid, but okay,
well,
whatever.
that would be the giant squid in the
The
room.
giant squid in the room,
Keep up,
right?
keep up. Right. What is an action potential? Basically, our question here is why does myelin help? Of course, we're going to discuss a lot more than that in this little segment here, but that's kind of going to be a question in the back of our heads, right? Or back of our minds. Why does myelin help this? And I feel like it's important before we walk through the specifics of an action potential to just set a couple ground rules as far as how these work, right? So generally speaking, action potentials are all or nothing.
Right.
Cannot have half of an action potential. You either have it or you don't. And all action potentials are the same size, right? They're all identical, right? The action potential individually that's responsible for, you know, carrying a stimulus of pain when you get a paper cut is the same size as the action potential coming from a huge gash or a car accident, right? The size of the action potential doesn't change.
Exact same.
It has more to do with the frequency of firing, which we'll talk about later, but we're all or nothing, and we're kind of one size fits all. Every action potential is the same size. We also only move in one direction. So we'll talk about why all these things are true, but I feel like it's important to set the ground rules for an action potential before we get into the specifics, because ultimately this same exact process happens in all of our neurons, right? It's pretty conserved, pretty shared.
And I think that like it's those foundational rules that again are also really testable on the MCAT, where they'll ask you these a lot of times like these sort of things are sort of overlooked, where yeah, we hear all or nothing all the time, but we don't recognize how significant that is. We say like, oh, how do we communicate nerves if they're all the same, or signals if every action potential is the same, right? It's recognizing these rules and then asking yourself, okay, if this must be true, why is that then? Yeah, why is that a pin prick versus if I chop my hand off, like if every action potential is the same, why is it that one hurts way more than the other? Right? So these are the sorts of questions. So I think you're totally right. Knowing those rules to begin with helps us to understand and ask questions even further. And so I think when we talk about the action potential, you can't talk about without talking about electrical signaling and like the pathway and all those things. But I think there are certain like terms and points we should be familiar with before we even talk about the action potential. Because I think a lot of the times the reason that, in my experience, a lot of people struggle with the action potential is we try to dive too quickly into it. We try to memorize all the things and we don't recognize the important voltages or potentials that are involved. So I think the first thing to recognize with the cells is the idea of the resting potential. right?
Mhm.
The resting potential is this when every neuron, right, at rest has this negative 70 resting potential. But it's very interesting when we talk about this resting potential. Like, okay, so at rest it's negative, but why? Why is
Mhm.
this the case? I know, Molly, you love talking about this. I'm going to leave the floor to you because I know you love this one.
I do. I love talking about action potentials as a story because
Hmm.
everyone loves a story,
Yeah.
right? I like to imagine that I, as a human being, right, or my arm, right,
Oh my goodness, the arm is again.
right, our hand and our arm as a neuron, right? I am a neuron and I am at rest.
Hmm.
Okay. There are things going on in my body at rest that are maintaining homeostasis of that neuron, if you want to call it
Hmm.
that, right? There are a couple things that are going to make me negative with respect to my environment. Keep in mind, these are all relative,
Hmm.
right? So how much more negative am I than my environment? It's equivalent to negative 70 millivolts of potential, okay? The main components of this, yes, we have molecules that are large, like DNA, that have negative charge. We have some
Hmm.
negatively charged proteins. It contributes a little bit. But the most important thing is actually the sodium potassium pump,
Hmm.
right? Sodium potassium pump is going to be a pump, meaning it's using ATP energy or using energy of some kind to force ions, in this case, against their concentration gradient, right?
Hmm.
Specifically, the sodium potassium pump is going to move sodium and potassium against their concentration gradients using ATP. We can use the 321 Nokia mnemonic here. If you haven't heard this, listen up. It's wonderful. 321 Nokia,
Hmm.
Nokia, tells you what the sodium potassium pump does. So
Hmm.
three for three sodium, which is Na, out. So three and no. If you wrote it like three,
Three,
two,
two,
one
one.
on
Okay.
one line, Nokia on the line underneath, this would be helpful and visual. Three, two, one, Nokia. Three, sodium, which is Na, out. Two, potassium, which is K, in. One, ATP used. So
Mm-hmm.
what this is going to do, this pump is going to be open to the intracellular environment. It's going to take three sodiums from the inside of the cell and then switch faces, face the outside, and push them out, right? We are concentrating sodium outside of the cell,
Hmm.
right? We are then taking two potassium from outside and moving it inside, and we're repeating. This process uses ATP, and what we're going to see is because we're losing three positive charge, we're
Hmm.
pumping three sodium out,
Right.
and only letting two
Two
potassium
back
in.
in. Right.
We are losing a positive charge every time we run the sodium potassium pump.
Right.
We are
Right.
becoming more negative.
Hmm.
It's also helping set up our concentration gradients that will be very important when we talk about things like depolarization, repolarization, hyperpolarization, all those things,
Hmm.
action potential stuff. But keep in
Hmm.
mind, I'm still at rest.
Right.
Nothing fancy is happening.
Hmm.
So the reason why I'm negative is because every time the sodium potassium pump runs, I lose a positive charge.
Hmm.
This is even further messed with based on the fact that we have little doors for
Hmm.
potassium to go wherever they want. They're called potassium leak channels.
Hmm.
If we know we're shoving potassium inside the cell and making it very concentrated there, again, imagine I took 10 of you listeners and shoved you in a very, very small room, and
That's
then I
not very
opened
nice,
the
but
door.
okay.
I know it's not very nice, but if I opened a door and said, hey, do what you want, you're obviously going to leave, right?
Hmm.
Why would you want to stay where you're so concentrated?
Hmm.
You're not going to, you're going to leave.
Hmm.
Those potassium leak channels are going to give potassium an opportunity to leave. Now we're losing even more negative
More
or more
positive.
positive charge,
Hmm.
right? So at rest, as a neuron, I am very negative because
Hmm.
of my sodium potassium channels
Hmm.
or sodium potassium pumps
Hmm.
mostly, and also my potassium leak channels.
Absolutely.
Um, and that's important. Again, most sodium is outside, most potassium is inside at rest.
Right, right. This is kind of a great frame to begin with because, like, this is our kind of starting point for the action potential. We're at negative 70, right? Again, yeah, that's kind of the really important information. Like, a whole bunch of sodium outside. We're at
Yep.
negative 70 and a whole bunch of potassium inside, right?
Yes.
This information is critical for when we talk about how an action potential even begins, right?
Mhm.
Because let's go back to our hand. Let's go back to the idea of our dendrites and our soma, right? Because let's say, like, let's start with we're first receiving the signal, right? Because the dendrites, those fingers on the soma of the neuron, right, is what receives the signal from other neurons, right?
Mhm.
And so when it receives that signal, like there are receptors, and we'll talk about that in a little bit about that whole process, right? When it receives that signal, it allows for gates to open, right?
Mhm.
When these gates open, right, the gates opening allows for ions to go into the cell. Because again, at first, at rest, we were at -70. But imagine if I get a signal from another neuron that's allowing more ions to come in or more ions to leave, right, that could possibly change my potential, right, as a result of that signal. Right. So this is where we kind of get two ideas coming in, which is what we call excitatory postsynaptic potentials or inhibitory postsynaptic potentials, where we were at -70, but in order to become activated, we need to reach this threshold, right? We need to reach this threshold potential of -55, right? Like millivolts. And so in
Or
order to be
positive.
like again, so we have to get a little bit more positive, right? We're at -70 to become activated. We need a little bit more positive. So in order to be activated, we need to let in more positive or let out more negative. And so these signals in the dendrites, channels will open and cause ions to flow, right? Either they'll be excitatory, allowing positive in, or they'll be inhibitory, allowing negative in or positive to leave, right? These are what we call graded potentials. Again, again, I know our conversation's on the action potential, but it's important that we make this difference because the MCAT will test you on this difference between a graded potential and an action potential. Because the graded potential isn't an action potential. It's these kind of localized changes in potential that could possibly trigger an action potential. Because we have to realize we have so many, again, I only have 5 fingers on my hand, right? Like I only have 5 fingers. But in
Wait,
reality,
really?
oh,
I have 8.
oh, you're right, giant squid. But the idea is that each soma has like hundreds of these dendrites, hundreds, and there are so many neurons sending signals, opening channels, that there are so many ions flowing in and out, so many localized graded potentials happening in the soma. Now, with all of these potentials happening, all of these different signals from different neurons, there has to be some place that we kind of interpret all of these signals. And that's the role of the axon hillock or the wrist, right? Where all of these graded potentials from all of these signals, all these net ions are going to kind of correspond or kind of coalesce at this axon hillock. And the net signal is what will decide if an action potential will fire or not, right?
Mhm.
Because, like we said before, there was this threshold potential of -55. If the result of all of these graded potentials maybe get up to -60, Molly, have we reached that -55 yet? No.
No.
So even if we've had all these signals, some ions leaving, some ions entering, at the axon hillock, we only reach -60, no action potential is going to fire. None at
Yeah.
all. And
I,
we're just going to go back. The sodium potassium pump's going to keep on going, and we're going to get back down to rest. Right.
yeah. I feel like it's really important to connect here the fact that all action potentials are the same size.
Right.
And we need, keep in mind, the brain especially is a very complex spider web of connections, right?
Mm-hmm.
We need to make sure if we're going to fire,
Gotta
we
be
need to make
certain.
sure we're firing for a reason, right? Because
Mm-hmm.
we're receiving all these all this information from all these other neurons that are connected in this complex web. I don't want to fire unless I'm supposed to fire, right?
Mm-hmm.
So we get all of these little inputs.
Mm-hmm.
We're trying to decide, is this enough? Is this
Mm-hmm.
enough, right? I
Right.
think it's important to consider how energetically expensive it
To
is
fire.
to fire.
Right.
That's part of it. But also the dangers of uncontrolled or random firing in the brain,
Mm. Mm-hmm.
right?
Mm-hmm.
Because if you have uncontrolled or random firing in the brain, you
Mm-hmm.
have a seizure, right? That's
Right.
what a seizure
Right.
is that uncontrolled firing. So
Mm-hmm.
we know we need this to be occurring exactly when necessary and
Mm-hmm.
not often else, right? Because we're going to exhaust our blood glucose. Keep in mind,
Mm-hmm.
the
If we
brain
kept
uses
following energy,
so
right?
much of the
Right.
body's energy that it
Mm-hmm.
comes in from the diet, but also the dangers of
Mm-hmm.
excessive signaling. And in fact, if you have excessive signaling, especially going on in the brain, it's perceived as anxiety as well. If it doesn't reach that threshold of randomness to cause a seizure, right? If you have excessive firing, that's anxiety. And we actually will talk about neurotransmitters, but there are neurotransmitters that its job is to produce one of those inhibitory postsynaptic potentials. These, as you said, their job is to prevent stimulation, prevent an action potential. If we need to go from -70 to -55, we actually need to become more positive, right? An inhibitory postsynaptic potential or inhibitory neurotransmitter is actually going to make us more negative. It wants to prevent an action potential.
Right. I.e. inhibitory, right?
Exactly. Yes. We'll talk about that. GABA is a big one in the brain, which we'll talk about. But it's important to identify that these neurons don't want to fire unless necessary, right? Very important. And they add. So we're taking in all this information and deciding with all of this holistically evaluated.
Love that.
I fire. I know. Applicable with med school applications being submitted soon.
Right.
Yes.
But yeah, like that interpretation of all those signals, because you are so right in saying, like, again, our brain is such an interconnected web. Yeah, absolutely. It's not just like as soon as one neuron tells this neuron to fire, it's going to fire. No, it has to take all of these stimuli into account.
Yes.
And that's why this axon hillock, this interpretation is so important, because it's a big deal to fire, right? So we have to make sure that we're certain.
Yes.
And so if we're not certain, it's not going to fire. But say we do get to that -55 millivolts. I guess the first question needs to be, why that number? Why -55? Because that's, again, I think that we always just say, yeah, -55, the next thing happens. But it becomes like, why is -55 so important, Molly?
So it's actually really, really cool. And it has to do with ion channels. There's a couple different types. We have ion channels that are gated by ligands. Basically, you need a molecule to bind, and then the ion channel opens. We'll talk about those later because that's actually how neurotransmitters work. But we have a ton of sodium ion channels that are actually, rather than opening in response to something binding to them, they're actually opening in response to a particular voltage of the cell.
Wow. Yeah.
-55 is the point at which those sodium ion channels, which are right now closed, open.
Right.
Now, it's important to think, again, when we thought of a door, the potassium leak channels, if there's a bunch of potassium in the cell and you open a door and say, go wherever you want, they're going to leave. Sodium is the reverse, right? Sodium is highly concentrated on the outside. Right? So very, very concentrated on the outside. If we reach that -55 millivolt threshold potential, those sodium ion channels open. Where's sodium going to go? It's going to come into the cell.
Right.
Positive things coming into the cell will make you more positive, and we're going to see a dramatic reversal of membrane potential. We started very negative, -70, we got up to -55. At -55, we're going to experience a fast moving wave of positive charges moving in. We call it depolarization because we were polarized at the beginning. We were negative, outside was positive. We are reversing that. We're becoming positive with respect to our environment. We're depolarizing that membrane. And now we're super positive.
Right.
That being said, we got to fix that, right?
We have just had this major upstroke, because I'm sure, if you're listening, right, you've seen an action potential before, right? You'll notice, like, as soon as you get that -55, now it's happening. And this is that all or nothing approach that we just talked about, right? As soon as you hit that -5, action potential is happening at full strength, right?
Yes.
It's not like halfway, like, oh, if I hit -40, like, or, oh, if I hit -56, like, no, -55, and we're happening. We're doing this thing. Right.
Yes.
And it's shooting up. It's shooting up to about +30 millivolts, right? That we're kind of shooting up to there. But then now that we are super, super positive, well, we don't want to stay that positive, right? Like, because, like, that's not very homeostatic to go from rest and then never go back. We need something like, whoa, they've totally changed up, right? We've got to kind of do something about this. And so something very important happens when you kind of upstroke up to this +30, because like we said, -55, that's when those sodium channels open. But now at positive 30, two kind of major, really important things happen, right? First, those sodium channels, as soon as you get to positive 30 millivolts, they sort of slam shut, right? The idea is that they're doors, right? Imagine them like the door in your room right now. They were open as a result of the negative 55, allowing sodium to go down its gradient inside the cell, causing that positive. But as soon as you get positive 30, think the door slams shut, right?
Yes.
But it doesn't just slam shut, it locks, right? This is kind of an important part, which we'll touch on why this is important in a bit, but it doesn't just close, it locks, right? And while there's another channel that we haven't really discussed, which are our voltage-gated potassium channels, right? These potassium channels were closed before. They weren't really doing much, but as soon as you got really up there, that positive 30, it's actually like a little bit before the positive 30, but for the MCAT, it's essentially positive 30, that these actually begin to open, right? But now these are just doors. Again, now with the potassium doors are open. But like we said before, there's a ton, as a result of the sodium potassium channels, there's a ton of potassium inside the cell. Well, if I open these doors, right, these voltage gated potassium channels, where's all this potassium going to go?
Well, think about it, right?
Right.
3, 2, 1, Nokia.
Hmm.
3, 2,
Hmm.
1, Nokia, right?
Hmm.
Ki.
Hmm.
Potassium is inside. Open a door. It's leaving.
Potassium's going to leave. All that positive charge is going to leave. And so this is what actually results in that downstroke, or what we call that repolarization. Because before, again, we just depolarized. Now we're trying to get repolarized to get back down there as a result of all this positive charge leaving. Again, those sodium channels are closed, but these potassium, all that potassium leaving, and we have this sharp downstroke, right? And so we go down and down and down, and we even reach our resting membrane potential. Wait, does that mean, oh, I'm back to resting? Am I good now? Like, am I done? Like, have I won? We're ready to go again?
Not quite.
Not quite, because there's something we haven't addressed here where we just messed up everything. Before the sodium was outside, now it's inside. Before the potassium was inside, now it's outside, right? Everything's mixed up right now. We have
Yeah.
to get back to kind of our resting membrane potential and our resting gradients. And so
Yes.
this is where we enter kind of that kind of third stage of like the action potential, which is the hyperpolarization phase, where we sort of overshoot that negative 70. We reached negative 70, that resting potential. Then we kept on going, right? We kept on going to about negative 80, negative 85. But this is where those sodium potassium channels, 321 Nokia, are really working to get everything back to normal. They are pumping that sodium back out, right? Because it all came in during the depolarization. We're pumping it back out. We're getting all that potassium back into the cell to get ready for the next action potential, right?
Yes.
And so
Yes.
this kind of like allows us to, so that sodium potassium pump gets us back from that hyperpolarization back up to that resting membrane potential. Once again, that negative 70.
Yes. So
And
I feel like it's important here
mhm,
to really emphasize that because I have so many students that are like, cool, by the time, again, we hyperpolarize, we're at resting, we get to threshold, we depolarize, we repolarize, we overshoot, which is almost like we got to threshold and then had like a inhibitory
mhm,
postsynaptic potential. We went too low and
mhm,
then we come back. Everyone's like, cool, we're back, we're ready to fire again. But
mhm,
again, think about you when you're inactive. Sodium is out, potassium is in. At
mhm,
the end of an action potential, sodium is in, potassium
mhm,
is out. You have the exact opposite. If you
mhm,
opened sodium channels, you're not going to get an action potential. Same thing with potassium channels because you haven't built that gradient. It costs money to do that, costs
mhm,
ATP. So yes, it's super important. You can't just automatically fire and fire and fire and fire and fire. You have to take a break, at least a brief one, so
mhm,
that the sodium potassium pump can fix the gradients
mhm,
before you can fire again. So
mhm.
super, super important.
Right.
And we call that period where we can't do anything. That's part of the refractory period.
Right, right. And in the refractory period, there's actually kind of two parts to that refractory period, right? There's the absolute refractory period, relative refractory period. And now to really understand the differentiation between them, I want to kind of recall back to what we said before, what happened to those sodium channels at positive 30, right? At positive 30 millivolts, those voltage-gated sodium channels didn't just close, but they locked, right?
Mhm.
So this is why the absolute refractory period is the thing. I want for those listening, the absolute refractory period is the kind of time where no matter what happens, no matter what graded potential occurs, this action potential is not firing, no matter what. Reason being is remember what actually triggered the action potential in the first place. It was those voltage gated sodium channels, where if you lock those voltage gated sodium channels, no matter
Nothing.
what happens, they're not going to open, right?
Yeah.
And so it's that result of that locking, and it's called the inactivation gate, that those sodium channels during the absolute refractory period are not going to open. Those sodium channels become unlocked at once they get back to resting membrane potential at about -70, they become unlocked once again. So it's kind of like once they first get back to that resting membrane potential, that's when you have finished the absolute refractory period because hey, they're unlocked now. So now you technically can fire again. But remember what happens after you first get back to that resting membrane potential. You hyperpolarize, right? You keep
Mhm.
on going to negative. This is what enters the relative refractory period. Because during the relative refractory period, you actually can still fire. But it's hard. You're instead of going from -70 to -55, you're now going from -80 or 85 to -55, right? The jump, that graded potential that you need to activate it is so much higher now. So while it is those sodium channels are unlocked, they can be activated. It's just a higher potential that you have to reach. And I
Yeah.
think kind of like understanding the two refractory periods is important because it's these refractory periods that allow, like I think one, that unidirectional idea, right? The idea that our neurons are only fire, our nerves only fire in one direction. The reason they don't go unidirect, both directions or bidirectionally, is because of this refractory period, where
Mhm.
that area of the neuron can't fire right after, so it can only keep on going forward, right?
Yeah.
And so again, I love the refractory period. Again, I took a whole class on this when I was in undergrad, so I was like, oh, this is so cool. This is the coolest. But yeah, just
Yeah.
I think it's cool.
I think at this point we could tie this back to myelin as well,
Mm-hmm.
right? If you think about why, our initial question is why is it so bad? Why do we have such significant symptoms when we lose our myelin? It's
Mm-hmm.
because yes, the myelin makes us, transmit neural impulses faster and it makes it less expensive, right?
Mm-hmm.
And it has to do with the fact that you basically have a long axon, right,
Mm-hmm.
covered by these little braces, these little insulating segments of
Mm-hmm.
essentially membrane. When you have that insulation, you don't have the flow of ions at that point, right? So we have these big chunks of essentially membrane insulation
Mm-hmm.
that impulse is insulated.
Mm-hmm.
It's not really diminishing. And because we
Mm-hmm.
only flow in one direction, it's fine. We though have periods or
Mm-hmm.
small little gaps between those myelin sheets,
Mm-hmm.
called the nodes of Ranvier, right? That we actually will have ion exchange to strengthen that signal to make sure even across long distances, we're not losing anything, right?
Mm-hmm.
So we have these insulating regions
Mm-hmm.
where we basically aren't spending any ATP to
Mm-hmm.
cover a lot of ground. And it's
Mm-hmm.
fast because we don't have to wait for ions to move and all that stuff. It's fast. It's
Mm-hmm.
just current carrying through a wire. It's like
Right.
lightning, right?
Right.
It's just traveling. Those nodes keep the signal strong, right? Which
Right.
is important. But if
Mm-hmm.
you lose your myelin, not only do you lose that insulating, like basically increase in your speed,
Mm-hmm.
but in order to propagate a neural signal, now we're losing ions along the entire length of the axon, which means our sodium potassium pump now has to fix very large ionic imbalances, and it's going to cost a lot of ATP, right?
Mm-hmm.
Which is not going to really work that well.
Right.
Um, so that's part of why demyelinating diseases are
Mm-hmm.
so important and why they
Right.
can have such a big impact on
Right.
the human body.
Right, yeah, super costful for the neuron at that point. And I like to add, like, especially when we talk about the nodes of Ranvier, I remember I had a professor that explained it super well. I was like in this huge lecture hall, and they were like, "Okay, everybody, do a wave." You know, like the wave at like sports stadiums. I always think hockey games, or you can think of like
Yeah.
football games. You know,
Mhm.
but like, you know how everybody does the wave, right? You know, it kind of travels across the scene. We did in the hall. And so
Uh-huh.
it took a little bit of time for everybody to do the wave. But then they said, okay, instead of everyone do the wave, only this row do the wave, and then moved over a few rows, and this row do the wave right after the next one. And which one do you think traveled faster across the room? The one where everyone had to do the wave, or the one where only certain rows had to do the wave?
Probably the certain rows.
Right, because they didn't have to wait for the people in between, right? This is the idea of why, like how I like to visualize like the idea of those nodes of Ranvier is like the name of that conduction is saltatory conduction, right? Between those nodes of Ranvier, like connecting, like sending that signal down. But how I kind of think about like why it travels so much faster, because it doesn't have to wait. It can just activate right away.
Yeah,
Yeah,
I like that.
yeah,
That's a good teacher. It's a good
right.
teacher that can put you
Oh,
in the zone for
what
it.
was her name? I remember her first name was Vivian. She was lovely.
Classic
Um,
Vivian,
Classic
honestly.
Viv. We love
Honestly,
Viv.
let's just channel our inner Vivian.
Let's just channel her inner Viv. She was a great teacher. She was actually great. Anyone who went to the University of Waterloo, during my time at least, knows Viv, and she was great. Um,
All one of you listening that
oh, well, one person.
very specific requirement.
But yeah, I think there's also like we can think of real world applications of this knowledge as well, like of this knowledge of the action potential, not just in the demyelination, but I'm sure all of us have had some experience with anesthetic, like local anesthetic, like lidocaine,
Yep.
right? I know
If you've had a tooth pulled or a cavity
yeah,
filled,
right.
definitely.
Right. I know I've had like I had 4 teeth pulled. Like I remember I had to like, yeah, I had braces and like my mouth, I just had too many teeth. So I had to get
Oh
four
my
teeth
gosh.
pulled. Yeah. But in order to pull the teeth, like the orthodontist had to inject, right? Lidocaine into my mouth and my entire mouth went numb. I lost all feeling in my mouth. But now we have to consider, what do you mean by losing feeling? That means my sensory neurons were not sending information to my brain anymore. We froze them using the lidocaine. But now we have to think, how do we think the lidocaine actually worked? What do you think, Molly?
Well, I mean, how do action potentials start? If we're saying that we don't have any sensation, keep
Hmm.
in mind sensation, right,
Hmm.
is communicated to the brain via action potentials, right?
Right.
Again, action potentials are used by all neurons. They're always the same size, right? It's all or
Hmm.
nothing, blah, blah, blah. If we have no signal from a region, right, that has been injected or, this
Hmm.
topical anesthetic has been applied, we are cutting off the ability to stimulate an action potential. And this
Hmm.
actually comes from lidocaine's mechanism of action, which is
Hmm.
to block voltage-gated sodium channels from opening.
Hmm.
If you can't open your voltage-gated ion channels for
Hmm.
sodium, you could have, right? You could have reached threshold, but
Hmm.
if you can't open those sodium channels, you can't trigger the action potential, right?
Absolutely.
You can't. Even if you're supposed to, right?
Hmm.
Even if your dentist is pulling teeth out of your
Right.
mouth,
Right.
right? Or something dramatic, it should hurt, but it's not.
Hmm.
Because even though your sensory receptors are trying to get that neuron to fire, right? It can't
It
because
can't.
you can't open those voltage gated sodium channels.
Right.
So
Right.
super, super cool. That's why you don't feel pain
Hmm.
when you have a local anesthetic.
Right. Which I think again is cool. And again, I always love bringing it back to this because a lot of the times, and we say this all the time, and people are probably gonna be sick of it, about the why we do this, like why we learn all of this stuff. This
The
stuff is
invention of it all.
interesting. This stuff is so interesting, right? Again, this
Yeah.
isn't a, oh, I have to learn about the neurons for like my MCAT, right? We get to because again, we can begin applying all this cool stuff that we know. Right.
Mm,
And I think again, when thinking about the applications of these things, especially for the MCAT, though, I think a lot of times you might see when it comes to the action potential, you'll get questions like, "What happens if we inhibit this channel? What do you think the impacts would be?" Right? Because I know we talked about this for the like when we talked about the kidneys, what we might see question wise for the endocrine system, what we might see question wise for the action potential. They love asking, say this happens where now this channel is now hyperactive or maybe underactive, or we inhibit this channel. We have to understand what each channel's impact is on the action potential in order to answer these questions. So
yeah.
again, I'm just nerding out, but understanding this stuff is the cool thing.
Yeah.
And I guess that kind of leads us well into the next portion, right? Which is why these action potentials are important. Because right now we've just talked about propagating a signal across an axon, right? So
Mhm.
now within that axon, it's gone from soma to
Mhm.
the end of the axon.
Mhm.
But now
Now
what?
what?
Like now, like, okay, big deal. It's gone from the soma to the end, right? Like, what happens now, Molly?
Yeah, I mean, the whole idea, again, we started off with, hi, I'm a neuron, I'm at rest. We talked
Mhm.
about the sodium potassium pump. We talked about the gradients we have. The whole idea is propagate the action potential,
Mhm.
but we're going to reach the elbow in our in our example, right?
Nice.
Like at the end of the axon, what happens? Like, what was the point of it all, right? The whole idea, like we said, is to pass the baton to the next neuron, right? Or at least to give it, throw your hat in the ring to get it to fire, right?
Mhm.
You want to stimulate the next neuron if you can. Again, not every stimulation event is going to cause an action potential. They'll cause those gradients, which may or may not trigger one. But your whole goal is, I have been activated by someone. I need to pass it on. It's almost like telephone. Did you ever play the game telephone?
See, I was the annoying kid that purposely changed it. Like
I knew
I
you
was the
were.
worst.
I could tell this about you,
I knew it. Yeah, I know.
right?
I'm getting a lot of flack for it now. I'd be the kid that thought I was so funny, like thinking, oh, this
Yeah.
is gonna change it so much. So
If,
yeah, but I know what you mean. Yep.
yeah. If you haven't played, it's basically you sit a bunch of typically kids in a line and you tell the first one something and their job is to say it to the next and 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 then the kid at the end has to say it out loud. It's kind of a similar situation, right? The whole point of hearing something with your dendrites is to turn around and say it to the next person,
Mhm.
right? You hear it with your dendrite ears and you
Or
say it
eyes.
with your axon terminal mouth, right? So we're
Interesting.
reaching,
That is the most interesting way I've ever heard it put, but okay.
yeah,
No, I like it. No, I like it. I get it. I know what you're, yeah, I get
right.
it.
The whole point is to tell the next neuron, right? So the idea is when you reach the axon terminal, which is where your neuron ends, you're going to reach this region, this open space between the end of you as a neuron and the beginning of the next neuron's dendrite, right? So you're going to have an axon terminal meeting a dendrite, not physically tethered,
Mhm.
right? It's not like a zap of electricity is going to go through and one is automatically going to be transmitted. There's a space between, and we call it the synapse or the
Mhm.
synaptic cleft, right? And the whole idea is this electrical signal traveling across the axon needs to release something that
Mhm.
can span that space and
Mhm.
cause potentially an action potential in the next one. And these are called neurotransmitters, right?
Mhm.
Neuro for nervous or nervous system, right? Transmitters for transmitting a signal between neurons.
Mhm.
So what we're going to notice is as this wave of positive charge, which is what an action potential is,
Mhm.
comes down to the axon terminal at the
Mhm.
end, what we're going to see is specialized calcium channels, which are also voltage gated, are
Mhm.
going to open. Now,
Mhm.
calcium is going to enter the
Mhm.
neuron because those channels will open as the action potential propagates. And specifically, the influx of calcium is
Mhm.
going to take these little vesicles, which I don't know why I always think of them on this like grocery bags. Like it's stuff, it's valuable stuff that's all nice and
Oh,
packaged together.
interesting. Okay.
If the cell was a giant grocery bag, right?
Okay.
Baby little membranes, right, full
Mhm.
of neurotransmitters are going to be triggered to be
Mhm.
released, right? Those
Mhm.
vesicles are going to fuse with the plasma membrane
Mhm.
when calcium enters, which occurs when the action potential propagates,
Mhm.
right? We're going to spill all of these neurotransmitters into that space
Mhm.
between what we call the presynaptic neuron, which is me, right, which is what we've been describing thus far, and
Mhm.
go to what we call the postsynaptic neuron. It's the next kid in the line of telephone, right?
Mhm.
Ultimately, neurotransmitters are going to span that cleft. They're going to bind to ligand gated ion channels, which are
Mhm.
typically going to allow sodium into the neuron. Typically, if we're talking about a excitatory neurotransmitter,
Mhm.
it'll let sodium in,
Mhm.
right? Which is going to increase our membrane potential,
Mhm.
possibly to the point of threshold, in which case, next neuron carries out that
Mhm.
action potential,
Right, right.
right?
Seeing these neurons like communicating, like knowing actually why, because I think a lot of us we like overlook, we're like, yeah, of course neurons communicate. They just send a signal, right? But we don't actually think about like, okay, how do they send that signal? What is the signal, right? It's not all action potential all the way through, like you said. It's not like they directly connect and send that action potential through. Getting
Mhm.
to the end, causing that calcium influx by opening those calcium doors, those voltage gated ones and causing those neurotransmitters. It's funny because I always think of those neurotransmitters like I don't know if you've ever seen like an electron micrograph. They're like these vesicles, these little like balls that are just jammed in the axon terminal. They are just so stuffed in that terminal, just ready to be released. They are so ready to go. And as soon as that calcium enters, they're like charged. Literally, like then
Yeah.
they just get released into the synapse, right? And so knowing that it's those neurotransmitters that are critical, because I'm sure we've all heard of neurotransmitters, serotonin, dopamine, all these really important things, acetylcholine, all these things, but that their role is simply to be in the synapse and then bind to a receptor on the other end. A big thing to note that's important, the neurotransmitters don't actually enter the next cell, the postsynaptic neuron, right? They bind to a receptor, cause that channel to open, right, and allow ions to flow in or out. But once it's finished binding, it gets released from the receptor, where it stays in the synapse. It doesn't go into the postsynaptic membrane, right?
Yeah. Hmm.
But now we want to consider, okay, we've released it, we've caused the signal, we've possibly led the ions in the postsynaptic membrane. Now we have all of this neurotransmitter in the synapse, right? Do we always want signals to be activating, Molly? Like, do we just always want them there?
No, right? That
No.
kind of goes against our homeostasis.
That's super not homeostatic. So we've got to do something with all these neurotransmitters. We've just released it. We worked so hard to release it, sending all this work to release them, but now we kind of got to clean them up a little bit. And there's sort of three kind of main mechanisms that we use to kind of get rid of the neurotransmitters that are in a synapse. Like the first one's pretty passive. Like they just diffuse, right? The synapse isn't an enclosed space. Like there's others, like they could just leave. Like they could just diffuse away, right? That's just randomly diffusion. Like peace out, right? That one's a little bit more passive.
Yeah.
Or, right, there's two other ways. The first way is that we can have enzymes in that synapse. Like a main one that we need to know for the MCAT is the acetylcholinesterase.
Mhm.
Guess what? It targets acetylcholine. And its job, we have enzymes that cleave the neurotransmitters. Like the neurotransmitters enter that synapse. So we have enzymes that can cleave those neurotransmitters and essentially inactivate them, right? So that
Mhm.
they can't keep on causing signal in the postsynaptic membrane, right? So we stop the signal there. Again, getting back to that homeostasis. And the kind of the third, and the one that I think really is important for the MCAT, right, is the reuptake, right? Because again, we just released these neurotransmitters into the synapse, caused the signal, we did our job. But maybe we want to bring it back. We want to bring it back into that terminal to be ready for the next time we need to release it. So we have these channels, like or these kind of receptors on the pre-synaptic membrane, right? Not on the post. Again, we had receptors on the post that cause the signal, right? But on the presynaptic neuron that's sort of ready for the neurotransmitter it just released. Because once then it's kind of just waiting for it. So once the neurotransmitter actually binds to that receptor, it's like, oh, you're back home, right? You did your
Yeah.
job, good job, right? And it kind of takes it and reuptakes it back into the cell and gets it ready for the next time to be released, right? And then so in these ways, using these three mechanisms, we can sort of like, okay, end what we just did, right? We caused the signal, now let's bring it back to homeostasis, bring it back to rest.
Yes,
Mhm.
absolutely. And I love that you bring this up because there are a couple really cool applications that we
Right.
could talk about, especially one, again, we're not talking a ton about, sort of like, I almost said pathopsychology, right? We're not talking about
Wow.
abnormal psych. But, you know, one of the most common treatments for depression
Mhm.
is what's called an SSRI, right?
Mhm.
Something like Prozac, right? Very, very commonly prescribed, right?
Mhm.
If you don't take it yourself, you probably, whether you know it or not, know someone who does take it, right?
Mhm.
Very, very common. You may not know this, but SSRI is selective serotonin reuptake inhibitor.
Mhm.
We haven't talked a ton about serotonin, right? But it is a neurotransmitter that is responsible for communicating a sense of well-being, right?
Mhm.
It's a mood regulator. It also does things like help regulate sleep and other things, but in part, its job is a mood regulator, right?
Mhm.
And generally promoting that sense of well-being. If we have a selective serotonin reuptake inhibitor, a
Mhm.
drug that is essentially inhibiting the reuptake of serotonin,
Mhm.
what do you think is going to happen to that individual that's taking this medication?
Right. Well, I'm kind of thinking about this. Well, that was one of the key mechanisms is reuptake to kind of get it out of the synapse. But if I'm
Mhm.
inhibiting the reuptake, well, now I'm just going to have so much serotonin in my synapse. Now it's not going to be cleared or as effectively anymore. The other two mechanisms might happen, but that reuptake isn't going to happen. So now that serotonin is going to spend much more time in the synapse. So much more opportunity to keep on binding to that receptor on the postsynaptic membrane and keep on exerting its effect. And like you said before, one of the main impacts of serotonin is that sense of well-being. And so this is why this medication, again, remind me, what is this medication prescribed for?
Depression.
Right? And so this is where that sense of well-being, you have more of that impact of serotonin to kind of aid with those symptoms of depression, right? And this is where, again, understanding that neurotransmitter pathway is important for applications. Again, I love bringing in medical applications, especially when it comes to medication. Personally, I love pharmacology. I think it's the most interesting thing ever. And so when you hear these like mechanisms that like, wait a minute, with our MCAT knowledge, we can understand this stuff. Right? And these make such a huge impact on individuals. And it's important to understand when you explain it to patients so they understand what they're taking, right?
Yeah, absolutely.
And I think talking about serotonin kind of leads us well to talk about other neurotransmitters that have very key roles, right? Because for the MCAT, like when you see certain neurotransmitters, like it can be a little bit overwhelming to try to memorize like everything a neurotransmitter does. The body is very efficient. It like if it has something, it doesn't like making a whole bunch of new molecules. Like if it can use the same molecule for multiple things, it will. So a lot of the times it's really not effective to try and memorize everything every one of these neurotransmitters does because like each one does a whole slew of impacts. But what's really important is to know like what's unique about this neurotransmitter? What is the thing that kind of separates this neurotransmitter from all the other ones? And so there's kind of some key ones to note. The first one, and I kind of mentioned it already before, was acetylcholine, right? Acetylcholine is a very, very important neurotransmitter, and where you're going to see it, where it's very unique, is for one, in somatic motor neurons. It's very much responsible for, your movement, right? For actually doing those somatic movements that we were talking about earlier, but as well, very involved in the parasympathetic response of the peripheral nerve of the autonomic nervous system. Where those parasympathetic neurons, those, rest and digest neurons, right, release acetylcholine to their targets in order to exert their effect, right? So acetylcholine has kind of these two major, when you see acetylcholine like in a passage, you should be thinking somatic motor neuron as well as parasympathetic, depending on the context, right? And there was actually a real world application of like this understanding that I know you really wanted to talk about. Molly, we actually discussed it a little bit before this, right?
I know. We actually both had different things that we wanted to connect to this, which was kind of fun, right? The real world application that we both like to talk about was botulinum toxin, right? Which is Botox, right? Which is a toxin, by the way, right? People injected it to their face and their arms and I don't know where else, right? It's a little toxin, right? And Botox acts, or botulinum toxin, it's just shoving those words together. It acts to block acetylcholine release, right? We talked about the different functions of acetylcholine, and there are a lot, but in partial, right, or in one of its effects, it activates voluntary or somatic motor neuron contraction, right? It activates skeletal muscles. If you inhibit ACH or acetylcholine release, it's going to cause paralysis, right? People inject it into their foreheads and their face and their eyes and whatever because it prevents you from being able to make facial expressions. You can't make facial expressions, you're not going to form wrinkles in those areas due to the repeated contraction of those muscles, right? So that's why if you see like a celebrity with a ton of Botox, they smile and you can't even like their whole face like it's the same.
Right, because it's all paralyzed. They've essentially
Paralyzed.
paralyzed their face using a toxin, which I think if you word it that way is shocking. Like
It's shocking.
that's such a shocking thing that we do, and we're just like, yeah, Botox, like 100%. No,
Botox.
we are
It's so
injecting a
complex.
paralysis toxin from a bacteria, right, botulinum, into your face so that you can't move your face anymore.
Yeah,
Wild idea, right?
Mhm.
And it's funny because when we brought this, like you brought that up, Molly, but then I remember learning about this like when in microbiome 2, like back in the day. And I remember what I took away from Botox is like Botox or botulinum toxin is the reason that you can't give babies honey or they can't eat honey before the age of 12. It's this because of this toxin is because
Well,
oh, sorry, 12 months, 12 months old.
yeah.
Actually, yeah, I should preface that. Yeah, I'm like, oh, if
One.
you're feeling
No,
you're
they're
11 year
1 year
old,
old.
you 1 year old or 12, I always think 12 months when you're that
12
young. And
months.
yeah, I'm like thinking 12 months. Yes. Thank you for
Yes,
imagine 11
just
year
to
old, like
clarify.
they're like,
Yeah.
stop to get in the way of honey. But yes, 12 months or 1 year old.
Yeah.
Because honey actually contains botulinum spores, right? This bacteria, like spores of this bacteria. And we as adults, we're fine because we have acid in our stomach, right? That when we eat the honey, right, it kills those botulinum. But babies don't, right? If you're less than a year old, they haven't actually developed the acid in their stomach. And so when they ingest it, those spores can become activated and you can have infant botulism, right? Or this paralysis as a result of this bacteria. And so this is where, right, before the age of 1, babies can't have honey. So fun fact about Botox and how it relates, how it relates to acetylcholine.
I love that.
Mhm.
Whenever we get together to talk about real world applications, it's always so interesting
Mhm.
because every instructor, like
Mhm.
our best friend Viv,
Yes, yes.
brings in different ways to
Right.
explain things. And it's really interesting to have our powwows because we are both instructors and have been
Right.
for so long. But
Right.
also we have had our own
So many,
instructors who have
so
shaped
many.
us. I love having these conversations with you, Mike, because it's just interesting to hear how many different ways there are to
Right.
connect this to the real world or to
Right.
explain something that you've learned again and again and again.
Right.
Um, as far as other neurotransmitters that you might want to be familiar with, there are quite a few. And again, just to reiterate, these neurotransmitters, if you Google like what are all the functions of acetylcholine?
Oh my goodness.
See things that are not related to muscular contraction, that are not related to parasympathetic signaling. There's a lot with attention and focus and other central nervous system activities. And I'll tell you, pretty much all of these on the list have something to do with mood, something to do with focus, or regulation of
Memory. Yeah.
attention and memory. Okay, so what we're going to do is highlight this is the thing that's unique to this neurotransmitter that if you see this discussed, you should think acetylcholine. Again, muscular contraction, acetylcholine, game over.
Right.
Parasympathetic nervous system activity, game over.
Acetylcholine. Game
Acetylcholine,
over.
right? When we see norepinephrine and epinephrine, right,
Mhm.
these are our neurotransmitters that we associate with the sympathetic nervous system. If you
Mhm.
see those discussed, sympathetic, right?
Mhm.
There's a little caveat here, right? And we're
Mhm.
not going to get into all the different neurons and
Pre-ganglionic, post-ganglionic. Yeah, no, no.
yeah, technically, sympathetic has acetylcholine too, right? So to see acetylcholine, you can't automatically say parasympathetic. Sympathetic neurons are acetylcholine, and then the ones that actually do something valuable, norepinephrine, epinephrine, right?
Mhm.
We know what the sympathetic nervous system does. We discussed that in depth earlier, right? Norepinephrine and epinephrine are the neurotransmitters that carry out that signaling,
Mhm.
which is super nice. And I know we were talking about how these trigger that stress response, right?
Mhm.
Short term, different from endocrine, which
Mhm.
we'll kind of touch on this again in a sec.
Mhm.
But to keep that momentum going, dopamine, which yes, creates pleasure, but so do things like endorphins, so can serotonin. Important about dopamine is motivation and
Mhm.
addiction.
Mhm.
Has to do with the brain's reward pathway. You've done
Right.
something good,
Mhm.
release dopamine, now you want to do it again. You want to do it
Right.
again and again and again and again,
Mhm.
right? You chase the things that give you dopamine because it's all about the reward
Mhm.
that drives motivation. Now,
Mhm.
dopamine is also very involved in motor coordination. And in
Mhm.
fact, if you talk about Parkinson's disease, right?
Mhm.
We've lost dopaminergic neurons, right? And you'll
Mhm.
see tremors a lot
Mhm.
in individuals with Parkinson's because it's not just the reward, it's not just the motivation and the
Mhm.
addiction side of it. It's also important in motor control.
Mhm.
Again, there's so many different
Oh
functions of
yeah.
chemicals.
Mhm.
Um, we've talked about serotonin, mood, sleep, emotional regulation.
Sure.
GABA is one of our
Mhm.
inhibitory
Like the
primary.
main. Yeah. Mhm.
We have glycine. That's less so in the brain, more so in the peripheral nervous system.
Mhm.
And then we've got glutamate, which is the primary excitatory neurotransmitter.
Right.
Those
And
probably are
the
the
main ones,
of the
right?
ones
Yeah.
you'll know. Yeah.
Yeah. Yeah. And again, to note, again, and this is something that like I know I personally overlooked for a while because I'm like, yeah, neurotransmitters, glycine, glutamate. Wait, those are amino acids, right?
Uh-huh.
Like we overlook a lot of times we're like, oh, these are neurotransmitters. No, no, no. These are amino acids that can
Yeah.
act like neurotransmitters. Yeah, yeah, glycine, yeah. Like glycine is an inhibitory, but like not really in the central nervous system, but glutamate, that very important neurotransmitter is an amino acid that's really excitatory. So I always like to add that, like to say, like, because I think we touched on that before about like the nervous system being super testable. Well, amino acids are in the exact same boat, in the exact same boat.
Yeah, they
So if you haven't already prioritized your amino acids, which you should be, like you need to know them yet like yesterday, right? let this be your sign to prioritize your amino acids.
We were talking about that. We were like, what topics? Because we opened this class this class, we teach too much.
Episode. Yeah, I
We
know.
talked this at the start of this episode about the fact that the nervous system is really the only asterisk, the only topic that can ride into all three science sections. All of them. Chem phys, psych soc, biochem, all of them. We were like, what other topics kind of ride that line? And the only other one that's kind of true is amino acids.
But that's still a nervous system, right?
You'll see amino acids in psych soc, like GABA, or like, not GABA, glycine and glutamate. But that's not tested on the fact that they're amino acids necessarily. You're not going to get like, oh,
still in
how
the context of a
combined,
nervous
peptide
system.
bond,
Sure.
right?
Yeah.
The names are there, but you're not really going to see amino acid protein content
Questions. Yeah.
super frequently in psych soc, right? Nervous system really is that bridge between all of the topics, which is why we couldn't not talk about it.
Like, especially after the endocrine system too, right?
I know.
And a cool like integration with the endocrine system. Something we chatted about last episode was the adrenal gland, right? We know we were talking about the kind of the slow response before, how we have like, if we get stressed, we have this longer response releasing those stress hormones. But the nervous system is this faster way, but the nervous system and endocrine system are actually really related. And I think a really great place where you can see that is in the adrenal gland, specifically in the adrenal medulla. Right? Kind of a callback to last episode where we talked about what's released in the adrenal medulla, the main mnemonic that we're thinking about. Amend. I was gonna like do a thing where like, okay, on three, Molly, 1, 2, 3, amend. But okay, whatever. But it's
ruined
fine.
it.
It's whatever.
Okay,
it's okay.
Rewind. No one has said anything. What were you talking about, Mike?
All right, that mnemonic that. All right, 3, 2, 1. Amend. Well, amazing, right? Which is that was so insane. That was so insane. But amend stands for adrenal medulla, adrenal medulla, epinephrine, norepinephrine, dopamine. Right. Yes. And we talked about pathways that result in release, like these axes before, hypothalamus, to pituitary to adrenal axes. But again, notice what we just mentioned comes from the adrenal medulla, the epinephrine and norepinephrine, which we just said were part of the sympathetic nervous response. So this is a cool integration where the nervous system actually synapses onto the adrenal medulla to cause the release of epinephrine, norepinephrine from the adrenal medulla, right? The adrenal medulla isn't controlled by the kind of this axis like the cortisol is, but rather, right, it's controlled by direct synapse onto the adrenal medulla. So I think that's kind of a cool integration of stuff that we talked about before, where the nervous system is actually like onto an adrenal gland to reduce these kind of amino acid hormones. So I just thought that was cool considering what we talked about last episode.
I agree. And if you haven't listened to that episode, I feel like, again, to call back all the way to the start, we weren't initially planning on talking about the nervous system, but you can't talk about them separately because they have such similar functions in many ways, but the mechanism through which they get there is very different, right? Nervous system and endocrine system are very different, and yet they are both trying to carry out bodily signals from place to place, right? They're very similar in many ways. The time course are different. The mechanism that they achieve their goal through, very different. But we had to talk about them together. They're very related. So if you didn't listen to that episode, go back one, check it out.
Go check it out.
It's our most recent before this one. But again, we kind of took a little detour. We've been talking about homeostasis. We've been building this idea of, different body systems and homeostasis and equilibria. We talked about kidneys and we talked about all of these good things, right? We were going to talk about endocrine. And then immediately after we were thinking, okay, more homeostasis. What else can we talk about? We had to talk about nervous system, as we've established. But our next topic in the lineup is going to feel a little different from nervous system, right? Very similar, though, with our initial momentum, which is homeostasis. We're going to talk acids, bases, right? In many ways, we are continuing with our same goal, though, of covering insanely high yield content that will be on your test.
Very much so.
Very much so. So next topic will be acids and bases. Get excited, you guys.
Probably one of my favorite topics. I know, like I teach, of course, we've taught you and I so long. I think acids, no. But when it comes to acid bases, like I know so many people have learned like how many times do we learn about acids and bases in undergrad? So many times. Like it's
Yeah.
so important. Yet I still feel that a lot of people don't come out of it actually understanding acids and bases.
I was
About
gonna
like
say,
right?
ask
Like they
me how
don't
many times I learned it, and then ask me how many times I understood it. Very
right?
different numbers.
Very different answer, right? Because and I think it's because we have this idea to like oh memorize my acids and bases. But I'm so looking forward to the next episode because like it's one of my favorite things to teach because like when it clicks and you understand why an acid is the way that it is, why a strong acid versus weak. I hope that people are excited because I think people will enjoy this one.
Yes, me
Mhm,
too.
mhm,
We're gonna have fun. Okay,
mhm.
well, go enjoy the rest of your morning or afternoon or evening or weekend or whenever we've caught you with this
Mhm,
episode. Happy studying, right?
mhm.
And we'll catch you in the next one for acids and bases.
See you
Thanks for
later,
listening,
everyone.
everyone.
Bye
Bye.
now.